Welcome back to the Facts Verse podcast. I'm your host, Tim Blaine. Today's guest is Dr. David Rasnick, a biochemist and chemist whose career has been defined by challenging mainstream scientific thinking. For decades, he's argued that cancer is driven primarily by chromosomal imbalance, known as aneuploidy, rather than by the gene mutations that have dominated cancer research for much of the last half century. His latest book, The Outsider's Advantage, a personal odyssey into the essence of cancer, combines his scientific theory with the story of his life and what he describes as being a scientific outsider. His work has attracted both devoted supporters and strong criticism, particularly for his views on HIV and AIDS and the public health. Today, we're going to explore his ideas, the evidence behind them, and the larger questions they raise about how science accepts or rejects new ways of thinking. Dr. Rasnick, welcome to the program.
It's a pleasure to be with you, and I'm glad we got together.
And first off, the wallpaper behind you is marvelous. I love that. It's like the best background we've had so far.
Well, the thing is I've been doing, since the book got published, I was doing like 3 or 4 of these things a day,
Yeah.
For a while, and she decided to come up with this thing back there, so I put it up.
That's great. That's great. It definitely makes it pop, which is great. So this book is kind of half memoir of like what it's like to be somebody who is treated in a certain way in the scientific world and what the ramifications are for your career, for your life, where you're living, all things like that. And then half getting into detail about this work that you've been doing over the last many years in this your theory of aneuploidy in cancer, and sort of the history of the thinking and who you got your who were the people who came before you, who you've worked with, and where you are now, and as well as where we should go in the future. So there's a lot, but it is, I will say, it's not some like weighty tome that I couldn't get through. I mean, it's that you condensed a lot into this book, which is impressive. Because and the nice, I'll get into the nuances of the book, but I wonder, going into writing this, did the memoir aspects of it come first or later, or while you were writing it?
It came later. I wrote a book for cancer researchers called The Chromosomal Imbalance Theory of Cancer, 2006, I guess it was. I forget the exact year. It had over 1,100 references, and it was very, very technical, and it was for those researchers, all right? Then I decided to tell the same story, but for the public at large, and say it in plain language, and it can be said in plain language. In fact, one of the first guys, Theodor Boveri, which we'll be talking about here in a little bit, he wrote a 59-page book back in 1914, plain language explanation of the aneuploidy theory of cancer. He had it pegged over 100 years ago, and then it got lost. We can go into those details at some point if you want to. But then a friend of mine, a journalist in New York, when I was writing the book, before I put my personal stuff in it, he said, "Dave, you need to put your story in there." And it didn't take me but seconds to click with that. I agreed with him completely. And fortunately, I hadn't gone into too much of the book at that point, And so I just told a story. And before, I was just going to tell the story of cancer. But then with all the stuff that went on with AIDS and the institutional problems and with the cancer, plus if it hadn't been for AIDS, I never would have joined up with Professor Peter Duesberg, and he and I would have never worked on the aneuploidy theory of cancer. And it was just Hollywood could not come up with the kind of story of my life and Peter and what we did. I mean, it was just this twisted turns and go this way and that way. And every one of those turns led to another adventure, and I could just go on and on and on about this, but my book is part of that adventure. Yeah.
Well, and it definitely explores, the torment that comes with, just being in the scientific community and just like having to deal with the larger institutions and even things like peer review and, just the powers that be and the money factors. And so, the reading it, there's definitely a sense of like feeling like the underdog as you're trying to push forward. But unlike something like, the first thing that popped in my head was the Rocky movies with boxing. Like, scientists are working on much larger and more important, if you will, things than winning some fights. I would imagine as you have gone along, I mean, do you ever find it hard to keep working because you feel this weight of these monolithic beings, pushing against you, or
No,
does that trigger you to work harder?
no, 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 no. My wife and I talked about that today, actually. Interestingly, uh, no, no, not until COVID, and then COVID, uh, especially after the injection period, 2021, and then, uh, then I started getting angry for the first time in my life, really. And I was angry at the institutions, at our government, at the global government, of everything that was going on. And now I said enough is enough, basically. Before, um, no, and my book, I don't, and that book has a lot in it, but believe me, my story is a lot more a lot bigger than that, but, I had to condense it, and
Sure.
make it there, but, yeah, now and I'm doing pretty well right now getting rid of that anger because it doesn't do any good, and my wife and I talked about that recently, and I said, I just gotta put that aside because it's not productive."
Yeah, I would imagine also just a function of as we all get older, we just kinda go, "Oh, yeah, I don't," that can happen. I feel like everybody I know as they mature just feels like, "Yeah, I was so angry about X, Y, and Z." Of course, when it is your actual career and your, like, larger things, we understand the anger. Um, we talked briefly right before we started recording. I thanked you because I was worried that this book was gonna be completely unmanageable for me as a non-scientist. I don't have a science background, but I was pleasantly surprised that I could read it and I could follow it. There was times when I was like, "Oh, okay, I
Oh,
gotta look
sure.
this up, I gotta look this up."
Oh,
And you even say there's a moment in it where you kind of go, "All right, for all of you non-science and medicine people, you might wanna skip this chapter because it's about to
yeah.
get dense," which is exactly what I did. Um, was it challenging to kinda keep that? Because also I would imagine you didn't want it to feel too light and too kind of, like, ephemeral for, like, this hard science that you wanted to put into it. How much back and forth was there about making it harder to read?
It was really easy.
Yeah.
Because you see, it's a story. It's not like I'm teaching a lesson.
Right.
Some of the chapters and the sub-chapters things that I got into the specifics of cancer, how it progresses, about what chromosomes. I wanted to tell a story that would resonate with people because it was like me. I mean, how did I learn about it? I didn't learn about it all at once, although I did have the classic light bulb moment, at one point. But these things are they're a journey. They literally are a journey. And the thing is that it was fascinating when I look back on it. And of course I always thought about it. My life's been fascinating as long as I can remember, all the way back to grammar school. Not fascinating in the sense that, oh, I did all these wonderful things. It was just a journey, you know, and it just kept changing and evolving. And I had I had 4 biotech companies, and they were all torpedoed, and primarily because of the AIDS stuff. Back in the mid '80s, I wanted to actually work on AIDS like all the other guys like me,
Right.
and a company I used to work at, Abbott Laboratories, came up with the first HIV protease inhibitors, an enzyme that HIV, relied on to replicate and everything. And that was my forte, coming up with enzyme inhibitors. And I started designing those things because I had a lot of experience in it already. All I did 4 classes of proteases. All I needed to know what class it was. Once they told me it was spiral protease, then I knew what the jaws look like, and then I could start designing inhibitors for these things. They're like tinker toys, and then I stopped it because Abbott had been working on this inhibitors for that type of enzyme for years, and I guess they had 2,000 of those inhibitors already in a bottle. But when I met an Abbott guy some years later at a conference, I asked him. He said, "No, 3,000." They had 3,000 in 1984.
Oh, wow.
When that came out. So,
Yeah.
This is a tree with 1,000 limbs, and
Right.
I don't want to just keep jabbering along, but I want to follow your lead,
Yeah,
and
yeah, of
help you
course.
understand stuff. But
Well,
I
yeah.
enjoy telling the story, so I have to tone myself down.
It's great. Well, you should enjoy it. I hope you enjoy it because it's definitely, as you said, it's an interesting one. I guess let's also, just for the sake of the people who haven't read this and aren't familiar, can you give us a little sort of like the layman's background on, like, okay, what is aneuploidy? How does it
Yes.
relate to cancer? Why is it that you're convinced that it is the cause of cancer as opposed to genetic mutations and things like that?
Right. Okay. We'll just start like where the book does. Back over 100 years ago, with David Hansemann. He was a German back in the late 1800s. And they had very good, high quality microscopes in those days. And then Theodor Boveri, also a German back in that period. Hansemann and Boveri knew each other. And Hansemann worked on cancer. That was his thing. And Boveri worked on development of organisms. Fertilized egg, how it progresses, goes this way and that way, net biology. Brilliant genius that he was. And of course, cancer was a developmental thing. And it had to do with cells. And he understood all of that part really easily. But, so he wasn't really a cancer researcher when he got started, but Hansemann was. And Hansemann, under a microscope, he had examined over 10,000 different cancer cells, over a period of time. And no two were alike. He took photographs of them, had good cameras on those microscopes in those days. He actually drew pictures, with pencil drawings of the cancer cells. And he had the normal cells of a certain type, whether it's your liver, your kidney, your blood cell, they look like photocopies of each other. They're very, very, very similar. All right. And the cancer cells, no two are alike. Your normal cells, we have about 20 to 40 trillion cells in our body, you know, and we have 46 chromosomes, two pairs of 23. 23 come from the mother, 23 come from the father. All right. And those 46 chromosomes in a lifetime stay put in your body after your cells, 20,000 to 40,000 cell divisions in a lifetime. And they have very few little mistakes. That's an astounding thing. And those 46 chromosomes stay in the normal cells. The cancer cells, they have an abnormal number, imbalanced number, and they just every time a cancer cell divides, an aneuploid cell divides, it scrambles. It's like shuffling a deck of cards. You never get the same combination of the chromosomes as the cells divide. And that's what Hansemann saw. He saw that with his microscope. So that was very, very good evidence that these chromosomes, the word chromosome was actually uh, pretty new in those days to call it chromosome. They called them other things, but then they started calling them chromosome. And he never saw two alike, and they're all different. And Boveri, like I say, he was talking. He was communicating with Hansemann. But Boveri was interested. He looked at it in terms of an imbalance, because when he looked at living things as how they progress, he understood that very, very well. And obviously, these cancer cells were never balanced. They were just always changing, all the time. No two are alike. To this day, there are no two cancer cells that are alike, that have the same combination of chromosomes. They break them up, they stitch them back together, and they almost always die, thank goodness, because they're damaged. All right. And so Boveri, uh, he did some extraordinary experiments, very, very simple experiments. Anybody could understand what he was doing. I even put him in my book in there, about how he talked about the consequences of these unbalanced chromosomes. And he spelled it out. He finished that book in 1914. He died the next year, in
Mm.
1915. Of course, that was during the World War II. World War One was 1914 and 50. And Boveri died, and it never took off. His chromosomal imbalance theory. Thank God he published that book, though. That he did, because we wouldn't know this. I wouldn't have known it. And then there was the Second World War and all the horrors in Germany. And then Watson and Crick discovered the double helical structure of DNA in 1953. And the United States was not the lead, was now the leader of the world, not Europe. And so they changed. Biology became molecular at that point, and it lost its trappings as really biology. And that switching to the DNA, people made it too important. And then they thought that the DNA was the instruction. They thought the genes were, were these things that I like to use this analogy. A lot of people think they don't even know this. Most people don't even know it. Not only the genome project, one of the genome projects, and a lot of them now have confirmed that different species. A lot of people, public doesn't know this. Humans and mice and a lot of other animals, we have
Yeah, the
99% of our
same. Yeah,
genes are identical.
that's wild.
You know that?
Yeah.
Identical. And something like 80% or so are in the same sequence in mice and humans. All right. So where is anybody writing a book? How do those same genes know to make a mouse or make a human or a tortoise
Yeah,
or
right?
anything else? All right. Well, it's because the thing is that the genes are not an instruction manual. All right. They just code for things like how to make, they're like a spelling that tells you how to make a protein, that protein. That's what that gene does. And I like to say that the genome is a biological dictionary. All right. It's a biological dictionary. In other words, think of the Oxford English Dictionary, which comes in 23 volumes, just like we have 23 chromosomes. That's why I like this analogy. Everything that was ever written in English, you'll find those words in the Oxford English Dictionary. Keep adding new ones to it all the time. All right. But guess what? You could study the Oxford English Dictionary or study the genome, your whole life, and you'll never find my book or Shakespeare play or
Right, yeah.
anything in the Oxford English Dictionary, you know?
Yeah.
And you study those genes your whole life, you'll never discover a mouse or a human or cancer. So gene mutations do not cause cancer. Let me tell you something else that the public is not aware of. But many of the mainstream cancer researchers over the past 10 years, and I even give presentations on this, I think I may have even quoted some of them in my book, I'm pretty sure I did, that acknowledge that cancer genes don't exist, that they don't cause cancer. All right. And more people are starting to talk in the mainstream scientists are starting to talk about chromosomes, but in connection with cancer genes.
Mhm.
They cannot do away with the gene, all the gene stuff. It's a $1 trillion industry. The genome approach to health and
Mhm.
cancer and changing all these things is totally worthless. Okay, now I'm going to pull on the brakes right here.
Yeah,
I,
yeah.
I, I could go on and on and on. I've gone a little bit too far on that one.
Well, that's great. But when it's interesting because this was gonna be another question of mine, which is that, like, it struck me, having read it and then kind of trying to poke around and do a little preliminary research before we spoke, was that, now more than ever before, the gap between what you're saying and what kind of like mainstream cancer research is saying is much smaller. I mean, I would imagine it seems like that your approach is, I know you're saying they haven't separated yet from genomic research, but like it seems like they're at the very least acknowledging and kind of trying to include chromosomal elements in their analysis too. Do you feel that's progress, or do you feel like there has to be a split from genomes genetics to really feel
It's not
successful?
progress. It's
No.
not progress. There's so many things wrong with it. They're just trying to keep the whole shooting match going. All the money coming in, the papers getting published, drugs being developed. And that they're just using these little techniques to just keep going, and that's why there's a lot more discussion about chromosomes, but not chromosomal imbalance. They don't talk about that at all. They talk about it in terms of the genes and gene mutations, about
Yeah.
how this gene on that chromosome does something, and another thing that is actually is considered scientific misconduct. You're supposed to. You learn this. We learned this in school. When you're working in a field, you are supposed like a specific thing like cancer or this enzyme or whatever. You have these citations, these references you put in the prior history, the prior literature to what you were doing. That's really how you even learn how what you want to do, what you're trying to work on is by the prior literature and what other people have done. And you want to extend it, expand on it, or refute it, or do whatever. But you cite the prior literature. All right. They still do that, but they never mention Peter Duesberger or me, and they haven't for years. You know, and these things are blatantly about chromosome. We were the chromosome people, chromosomal associated. Well, Theodor Boveri, I cite Theodor Boveri, Hansemann,
Right.
Papa Nicolau. That's how you're supposed to do science. All right. And here, Peter Duesberg and Dave Rasnick, uh, we proved Theodor Boveri's chromosomal imbalance. We proved it experimentally. Peter did the experimental stuff. I covered a lot of it in the book. Of course, we have the scientific papers that we published on that, and the book that was done. And the theoretical and the experimental work, and we crossed all the T's and dotted all the I's that there is. We've explained everything that is known about cancer, the time course, why gene mutations can't do anything, how the imbalance, why cancer is slow progressing, and the chromosomal imbalance is one of the reasons. There's not a single thing about cancer that I'm aware of. Doesn't mean we've covered everything, but everything that I know of, we have covered it,
Gotcha.
and it fits in perfectly with the theory. That's how you prove something in science.
Right. Right. Am I correct in saying that the mainstream cancer research acknowledges and agrees with you on in terms of like the existence of aneuploidy and, like that it's more like they're saying you're saying it's caused by aneuploidy. They're saying we have aneuploidy, but that's not the cause. Is that sort of the
Yes.
major? Okay. So that's the disconnect. Are there people who are either approaching you or doing independent research, trying to sort of do exactly what you're doing to sort of reproduce it? Are you getting people? Are you getting converts?
Oh, people have done well. Not anymore. I'm not in the lab.
Oh
I'm
yeah, yeah. You're
retired now.
writing now, yeah.
But I mean, people have been doing this. I even have it in my book. People had discovered the chromosomal imbalance theory of cancer. Boveri was one. Papa Nicolau did. I forget the other guys' names that I mentioned in there. They published papers. They didn't go anywhere. It didn't take off. But you see, these things were independent. And these other people didn't even know those other people. They
Right,
discovered it on their own, and
right,
that's sort of like independent confirmation. You know, and then Peter and I, we didn't invent it. We rediscovered it. You
right.
know, I mean, we knew about it. We knew about Hansemann and Boveri and stuff, so we didn't discover it in that sense. But we proved it. That was our contribution. We proved Theodor Boveri's aneuploidy theory of cancer, and then extended it far beyond that.
I mean, it sounds like one of the reasons you're saying, in addition to the money aspect and all the sort of political and whatever, that the hanging on to it's all genes, it's oncogenes, it's this is almost because like that was the loudest voice in the room, and then it snowballed, and then like no one goes back and questions the initial stuff. Like you have this foundation of it's all genetics, and then no one thinks to even what if our first thing is wrong?
Well, it's control. You see, I learned about this control, institutional control, goes way, way back. People have written books about institutional control, and it seems like it just keeps getting forgotten, and new people discover it and write about it. I saw this, learned about the institutional control with AIDS stuff that came out. Remember, I said back in 1984, before we didn't, I was just a chemist. I didn't know what this AIDS stuff was. I lived in San Francisco, the Castro district. The gay guys they're the ones that had the AIDS, but people across the street, they didn't. And that wasn't my thing. Other than, okay, it exists and I'm right here with it and we can talk about it and everything. And then when Robert Gallo was brought out there to talk about his HTLV3, which later became HIV, and all that scandal, we could get into that if you wanted to, but it was a pack of lies. That whole thing was a pack of lies and fraud. And but then we didn't know it then that it was a pack of lies and fraud. Uh, but we got, oh, great. Now we know what it is. Like I say, I want to make my enzyme inhibitors for this type of virus and thing. Uh, and, but one of the things when about a year after that, it's supposed to be a contagious thing. And you look at all the different diseases, the different people, hemophiliacs don't get the gay guys' diseases, and IV drug users get their diseases. And if you're not gay or using these drugs, you don't get AIDS, you know? So where is it? It didn't behave like a contagious disease, and it was very clear. I think I might have. I don't know if I put it in that book or not, um, but I've written about it. Uh, it was clear that AIDS was not a contagious disease early on. I wasn't the only one to think that. A lot. The CDC knew as early. I learned about this later. The CDC knew as early as 1982 that AIDS was a recreational drug disease in the gay community. They knew it was not contagious. Of course, you won't find that now. It's pretty much erased. But the CDC understood that as early as 1982. And then 1984 is when Robert Gallo was brought out there to turn almost 30 some odd diseases that have nothing to do with each other into a contagious disease. By the AIDS-defining diseases became AIDS. They weren't AIDS before, but if you had antibodies to HIV, now you no longer had this disease or that disease, but now you had AIDS. You used to have Kaposi sarcoma, you had AIDS, for example. And that was that didn't make any sense. And there was it was obvious to Peter Duesberg, plus he knew about retroviruses. It was obvious to a lot of people early on that it was a pile of crap. All right. There was something seriously wrong here.
And
But
let me just stop here just for an explanation.
sure,
When you say it wasn't a contagious disease and it was an intravenous drug disease, so like obviously it was spreading. Are you saying that it was not contagious from person to person, only through
it's
injecting?
it's not an infectious disease. It
Okay.
was not caused by a virus or a contagious disease.
Okay.
Uh, yeah, we can get into those details if you want to. The important thing that I want to say right here is one of my early light bulb moments. We had this Bay Area group that met once a month. We'd go to a seminar at University of California, San Francisco, and then go out to a Chinese restaurant after. We all talked together, biotech guys, academic people, and we'd form occasional collaboration, and we chit chat about once a month. And when I wanted to bring up the point that I was doubting that AIDS was contagious, it didn't behave like a contagious disease. Nobody would talk to me. You know? Listen to me. Everybody, you're on your program, I love to talk. Scientists love to talk.
Yeah.
And when scientists shut up, something's seriously wrong. And that shook me, man. I didn't know what to do. That's when I started going to the bookstores in the gay areas in San Francisco, and the wonderful bookstores, tons and tons of books. And I was reading about all this stuff in AIDS and learning about it. And then I think it was 87 or 86, somewhere anywhere. Something was really, really wrong with the whole thing. I didn't know who Peter Duesberg was at that time, but in 1987, Peter Duesberg, world authority on retroviruses, HIV is supposedly a retrovirus, and he wrote an invited article in Cancer Research in 1987, and it was like a legal document, point for point for point for point how HIV could not cause AIDS or anything else. It was a retrovirus, which he's an authority on, you know, and it was basically a pack of lies. I mean, he didn't say it was a pack of lies, but all of his evidence did. And of course, he was a member of the National Academy of Science. He had that outstanding investigators grant, which was what really everybody wanted those because it took every 7 years, not like every year or every other year you have to apply for new money. Give him like this long window. Well, that all disappeared overnight with his 1987 paper. He couldn't get any more graduate students, and they kept treating him harder and harder
Mhm.
as things went on. And then when I joined up with Peter, you know, it rubbed off on me too, but in a good way, you know. And we worked together. I think I put a little bit of that in my book. We worked together on AIDS, alternated between that and cancer. And Peter went to Germany the next year, and I stayed back at Berkeley with him at Berkeley, studying aneuploidy at the about a week or so before Peter left to go to Germany. I asked him, "What are you going to do in Germany?" And he said he was going to work on. Can't remember the exact words, but he was going to work on aneuploidy theory of cancer, experimental with animals. That's what he did. And I didn't know. I don't even think I knew the word aneuploidy at that time, you know. And I worked, with maybe 20 different enzymes, they come from different genes, 20 different genes or whatever. And we got 20,000 genes and Peter on all these chromosomes. And I was trying to think, what's he going to do with these looking with these chromosomes? He told me about the aneuploidy. And I said, how am I going to figure that out with all those 20,000? And then I asked Peter, I said, you mean it's just numbers? Just the sheer numbers of these things? And he said, yes. I went, wow. Peter went to Germany to do the animal studies, experiments, and I went to the library. I read everything I could on aneuploidy and chromosomes, and and on and on and on and on. And you can tell I learned a lot because I put it in my book.
Yeah.
A lot of that stuff that I learned in there.
Can you
Okay, I gave
talk
you a
about
question.
yeah, so, well, can you talk a little bit about metabolic control analysis, or MCA, and how it relates to what you're talking about?
Yeah, metabolic control analysis was uh, Kexer and Burns. These were the two guys that invented metabolic control analysis. And Kexer himself died the year before I joined up with Peter Duisberg, so I didn't get to know him. But I came across it in my study when I was studying stuff about chromosomes when Peter was in Germany. Uh, I, I know a lot of enzyme kinetics because I did that, and you look at these things, and I came across Kexer and Burns papers on metabolic control analysis. And metabolic control analysis. Let me give it an analogy for the people. Like in essence, it's a way of looking at the entirety of a living thing, whatever level you want to, at the cellular level, at the organ level, at the whole organism level, or whatever. You know, all of the constituents together. You're taking them into consideration, and you're trying to follow how they work, how that ecology is. Well, it's sort of like an economy. In economy, so living things are very, very stable, and economies, for the most part, are very, very stable. Year in and year out, they'll have these little rises, little dips, or whatever. And you got these hundreds of $1 billion economy or $1 trillion economy or whatever, and it goes up here, like moves around a little bit. But one reason it is so stable is because you've got all these people working together. You have consumers and producers, but all consumers are producers, producers are consumers, and think of them like the enzyme. The enzymes are like that in our body. Like the substrate comes in, an enzyme does something to it, and the products of that reaction go out, and other enzymes do something with that, build things or tear things down or whatever. The life is a biological economy. And we have the human economy, where you have people that produce this and consume that, and their products go over here, and somebody else uses it. It's just like a huge, huge economy. The flow of goods and services and money. All right, that's metabolic control analysis is a biological economy, and the analysis of those flows, going through living things. And you can have these big catastrophes like a crash or a boom or whatever in an economy. And you can have the same kind of crashes in living things. And Kexer and Burns were able to analyze that dynamics, like economists analyze the economy. You know, well, he was doing the same sort of. They were doing the same sort of thing on this dynamic with the enzymes. Primarily about enzyme reactions, because the enzymes are sort of the little machines that they're sort of like you and me. I make this and then sell it to you, or something like. Well, that's what the enzymes do. They move these things around. They build things, and they had a theory, metabolic control analysis, where they could connect them all together. Now that's astounding when you think about it. How can they do that?
Yeah.
And then I saw the simplicity, like the simplicity of the economy. You don't have to know the details about every little thing. You just need to know the overall flow. You don't have to
Yeah.
worry about every little person that put that nail in or took that screw out or whatever. You just look at the overall flow. And that's what they did with biology. Now, they did it on a theoretical basis. They didn't have the techniques to do it on an experimental level at the same way. But they were able to get up to scores of reactions, even up to hundreds of reactions, where they could do it in the laboratory and get each one of the little components, and they put part of their equation, and then you do these tests. What their theory would say, let's say on this collection of enzyme reactions that produce this part of your body, all right? And then their theory would tell them what to expect if you change the activity of this thing going into the thing coming. How will that affect the overall outcome? Right? And their theory told them that you can have large changes in individual things in a large complex system that'll go totally undetectable as far as the big system it is. I like to use the analogy, as we have this multi-trillion-dollar economy. If the president lost his $3 billion overnight, it wouldn't show up. It would not be a blip in the economy. Or if he doubled it, it would not be a blip in the economy. A huge, huge thing for him and the people close to
Right.
him, but zero in the economy. And that's what metabolic control analysis does with biology. It allows you to look at the whole entire anything in biology, like I said, a cell, a tissue, a whole organism, even an ecosystem. All
Mhm.
right. And his figure in one of those his articles or his paper, I looked at it and then I got it, you know, I don't want to go into those details right here. The picture does much better job of that.
Yeah.
And then I realized it, and then I started applying metabolic control analysis to cancer because, and it turns out so much easier to do it on cancer than it is with a whole ecosystem. If you compare one system to another one, it is so much easier than taking one system and looking at all the details of just that one.
Mhm.
But if you compare one with the other one and you see how they flow, it's so much easier to do the calculation. And that was my big contribution. And I put that in my book in that appendix in there to try to explain it about how and theoretically when you look at it, the theory says that you cannot have a massive change in a biological system like aneuploidy if a few genes are disrupted or even a few hundred genes are disrupted. Won't even show up, you see.
Mhm.
There's no, it just has no effect because I can't explain it here orally. You have
Right, right. No, yeah. But
right.
it's I see what you mean in terms of like, the genes in that case are the president and his $3 billion. They just don't have a
Yeah,
blip. And
right.
then, uh, uh, okay. So that
But
yeah, go ahead.
but it's not the magnitude of individual changes. It's the fraction of the whole thing that changes. In other words, if you have like a 5% or 10% change in the economy, you know, in the whole economy, that's the fraction of the economy. That's like if you have a 1,000% change in this billionaire's income, or has no effect on the economy. But if you have like a 10% change in the economy as it goes up or goes down, it has a massive effect on the economy as a whole. And if you and the same thing works with aneuploidy. That you get all of your enzymes in there, they're out of balance, and you get 30-40%. Typically, typical cancers have on average about a 70% imbalance excess of chromosomes, and they're not balanced at all. 70% increase. All right. In these things, it disrupts the whole activity of each one of those individual cells, and they almost always die. It's the fraction of the imbalance that leads to cancer and the aneuploidy. Okay, enough of that. I've talked
This is
myself to
great.
death
No, I
on that.
mean, this is why we have you on. This is, nobody wants to hear my theories. Um, so then what okay, so then but there are specific carcinogens that are then creating chromosomal imbalance or like how does it
Yes,
start in the average person?
yeah. All carcinogens. Something that causes cancer causes aneuploidy.
Mhm.
All right, and let's take asbestos. Asbestos. It's just a mineral basically. It's just the tiny little needles. They're chemically unreactive. They don't do anything other than what happens is these tiny little fibers can get inside your cell, and then you have this when your cells divide, when your cells divide, you double your 46 chromosomes, all right, such that when the cell divides, 46 will go to the new cell A and the new cell B, all right? And those chromosomes line up, they get really symmetrical, and the little proteins, fibers that pull them apart during cell division. They pull them apart like that. Okay. The fibers, the asbestos fiber comes in there and gets intermixed with those little fibers that pull the chromosomes apart. So now they're not balanced anymore. One cell will get too many chromosomes, and one
Mm.
cell will get too few. That and every carcinogen causes chromosomal imbalance. You will not get cancer without chromosomal imbalance. The chemical carcinogens do it, the physical carcinogens do it. That's what X-ray does. X-ray destroys the ability to have a balanced set of chromosomes during cell division. Every carcinogen that has been tested, that's what they do. They disrupt mitosis,
Mhm.
the ability
Yeah.
to have an even separation of chromosomes. That's what a carcinogen is.
Is there any room for your theory and a genetic theory to be true at the same time? Like, could they
No,
no, it's just like,
no,
no, this has to be one or the other.
no. And even the gene mutation people, the big shots. I quote some of them in my book, and when I give presentations, they know it. They have not come up with one oncogene, not one cancer gene, and all of this stuff. They admit it now, to their colleagues and in their literature. The public doesn't get wind of it, doesn't hear a word of it. That's why I put it in my book, and I use their own words. I cite them where they got it, where I got it from.
Yeah.
They know it. They're not stupid.
I
You
just
know?
go then, okay, there has to be some reason why people have latched onto the notion of oncogenes if no one's been able to find it, and like, is it strictly political and money driven? Like, what's your
It,
take on, like, why is it so strongly
oh,
held?
it's part of the. Oh, I this that's goes back to what I was going to get to, and I never did because I was climbing the other little limb on a tree. About the institutionals are so corrupt. They're controlled. A guy wrote a book about it. I can't remember his name. It's on my website homepage there. I can't remember what it's called, but if you download his book about the corruption of societies, and the more professional you are, an MD, a PhD, or big money, or whatever, the higher up you go in the ladder, there's more pressure put on you, especially medical students. People want to get in medical school. There's pressure put on these young people at the very beginning, high school or whatever, get the right grades, do this and do that, don't make any mistakes. You got to, and then they follow this all along. They do what they're told, to try to get into medical school. All of professions are this way, one way or another. Medical profession is probably the worst. And I saw it when I was at UC Berkeley with Peter there. Also saw it when I was at Abbott Laboratories back in the late 70s about the pressure to conform. You're just one person here, and you got this huge infrastructure, and everybody's conforming, and you just follow the thing. Keep your mouth shut, basically. And that's what a lot of them did at UC Berkeley. Peter's colleagues, other professors that were full professors, they could have spoke out if they wanted to. I was there in Peter's lab when they came in and were talking to Peter about the AIDS stuff. They said, Peter, I'm with you. I agree with you, but I can't go public. I can't support you, like that. And other people, the younger ones, these were people who already had their tenure. Then the younger ones said, well, Peter just doesn't know how to play the game. I mean, they're faculty, but they're not tenured yet. And that's what goes on. This pressure. If you want to keep your job and not get fired, or like Peter, totally lose everything he had, and those examples of what you do to the really visible people, extremely capable people like Peter did. Remember, National Academy of Sciences had the Outstanding Investigators Grant, the World Authority on Retroviruses, and that sort of thing. And then when he wrote that article against HIV, he lost it all, you know. It was a blessing, though. Like I said, it was a blessing. All of these crazy things that happened in my life, like my 4 biotech companies that got torpedoed because of my AIDS stuff and Peter. But we got together and we worked together, worked on cancer, AIDS, a lot of other things. So
Yeah.
to
So
me, it was,
with your theory, what are the treatments then? How should we be handling cancer?
uh,
Do we know that?
yeah, I mean, cancer, it's been known for a long, long time. Cancer is not, it's been around a long, long time, but it has accelerated, exploded in advanced populations, where you can produce more carcinogens, and cancer rates just keeps going up and up and up and up, and it's accelerating right now since COVID stuff, but not because of any virus or anything. And I forgot where I got derailed. What was that? What led to the question?
What are we gonna do? How do we treat cancer? How do we
Oh yeah,
use your
yeah,
theory to
the cure. Yeah. Okay, so the thing that people knew, there was this. What was the guy's name? Coley, William Coley. He was a physician in New York in the late 18th century, 19th century, early 20th century. And he was a physician working in a hospital in New York. And there was this guy who had a cancer, a sarcoma on his jaw. And he kept coming in, and this guy named Ewing would always perform a surgery on it, make it go away. But he kept coming back, coming back. And then one day, this guy, he had a high spiking fever. He had a bacterial infection of some sort, and he got this huge, huge fever, really high temperature. And almost immediately, the cancer went away. It disappeared, you know. And and then Coley spent, I think, I can't remember, 6 or 7 years trying to find this guy, see if he was still alive after that miraculous thing, and he was. And the only scar he had was where the Ewing kept cutting off the cancer, but other than that, he looked fine. And so he came up, he took that seriously. He observed something, he saw it, he said, "This is real." So he started developing his mixed bacterial toxins. They used to call them Coley toxins, where he would infect cancer patients with these bacteria, the ones that would cause these high spiking fevers. And they worked. Not 100%, but to this day, his daughter wrote a book about it, went all over the world and collected the data on these Coley toxin things, and and it's still being used in Europe a lot, and in Mexico, but only rarely in the United States, and only in conjunction with chemotherapy. So it's almost never used there. And the survival rate that she documented in her books, the 5-year survival rate of people that took Coley's toxins or mixed bacterial toxins was about 50-55%. Guess what this 5-year survival rate is on
on
chemotherapy
chemo
alone?
alone? Yeah, oof.
2 to 3%
Wow.
to this day. 2 to 3%.
For all cancers sort of like
Yeah,
put
as
together?
a group combined.
Mhm.
All right? They vary from cancer to
Sure,
cancer.
right.
There's no doubt about that. But when you combine, all right. So that's one thing. All right, and it makes me wonder. Think about it. A lot of places like the American Indians, the people in Scandinavia, they have these sweat lodges, and they have all these thermal things. You know, and I was wondering. I wonder what their cancer rates are, or how they got started doing that. And the other thing is that this is interesting. I mean, this is a tree with 1,000 things like measles, mumps, and chicken pox. This has been documented. It's known. If you go through those things, they prepare you to fight cancer. I've had all three. I'm old enough.
Hmm.
You didn't have those injections in those days.
Right, yeah.
You know, and and people that did not get those, they have a higher risk. Which are mostly people. Most everybody gets injections these days.
Yeah, I was
Have
gonna
a
say there's not
higher.
much of a group that doesn't at this point, yeah.
No, 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 and it's exploding since you might have heard of turbo cancer as a thing. I don't know if you've heard that,
Not
but in my
before your
book,
book, I said yeah.
yeah, I never heard. I'm a cancer researcher, and all the other cancer people, even though clinicians, never heard of that phrase before. Uh, but it's clearly there, and it's increasing, but doesn't get as much public play right now. So, as a cancer researcher, I was curious about this. I looked it up in the literature. It's easy with EndNote. You can search for the scientific literature all over the world. And I thought, there was no turbo cancer or rapid progressing or whatever it was. And finally, I found the term hyperprogressive disease in a paper that was published. The first paper where that term was, it was 2017, and where And hyperprogressive disease and turbo cancer that we're seeing now are indistinguishable from each other in the sense of how rapidly these cancers progress. The hyperprogressive disease, these people they were cancer patients. And I forget the particular type of cancer they were having. And they came up with these new drugs called immune checkpoint inhibitors. They're all the rage right now, immune checkpoint inhibitors. And depending on the paper, I keep looking at all these papers, over almost 300 papers I was able to download on this hyperprogressive disease. And they there would be anywhere from like 5% of the people that took these immune checkpoint inhibitors that were already cancer patients, anywhere from 5 to 70 or 80% of the people would get hyperprogressive disease, you know. And they're still publishing this in the literature. The thing that really annoys me, maybe I should use the word angers me, is that the publications are basically, they're just people who are reporting new cases of it, like we're doing this study, but you don't see, to see how well these things, but you don't see, I haven't seen any of people who are trying to understand why this hyper progressive disease is coming about with people that have cancer, and you're giving them anti-cancer drugs. Wouldn't you want to know what's behind that?
Mm.
You know?
Yeah.
Well, I would. Well, there were a couple of curious people, all right, and they noticed we have these antibodies in us, the IgG1, IgG2, IgG3, IgG4. Two of these IgGs are like the accelerator of your immune system, and the other two are like the brakes. If you get an infection, the accelerator goes in there, turns it up so that your immune system will fix you, right? And then the brake comes in there to shut it down, because your immune system is very destructive. It can be very destructive because it's destroying the stuff that's in
Sure.
your body.
Yeah.
Right? And the IgG4, which is the lowest level, anywhere from 0 to maybe 0.04% of your IgGs in your body when you're healthy, it's really low like that. It went up 700-fold in people that were getting these immune checkpoint inhibitors. In other words, it was shutting down your immune system's ability to eliminate cancer. And the cancer then would grow like it was in a petri
Yeah.
dish in a
Yeah.
laboratory. All right. And then I looked up the same thing with the turbo cancer folks, the people who were most of these people. I remember when I first heard about it, and since I knew a lot about cancer, I realized they didn't get cancer immediately. They probably already had it, and it was just released. This is
Mhm.
before I knew about hyperprogressive disease, but when I understood that, I said, no, they already had cancer, so something accelerated that cancer. I looked into that, and also some people were looking into it, and the exact same phenomenon they saw when the people got it injected with the SARS-CoV-2 uh, injections, that they had massively elevated levels of IgG4, just like the cancer patients. The ones that were receiving the immune checkpoint inhibitor, IgG4 went way up. The people
Mhm.
that got the SARS-CoV-2 injections and then they got the turbo cancer, the IgG4s went way, way up. Same phenomenon.
Yeah. Is there so, like, where does chemotherapy fit for you in the future? Can you be selective with it, or is it just sort of like, that's
Now,
a toxin that's just gotta be out?
there's nothing good about it.
Okay.
They're toxic. In fact, chemotherapy, I think, is the sixth leading cause of cancer. Radiation, even higher than that. I mean, they cause aneuploidy. There's nothing. I cannot see a single benefit of chemotherapy or
Mhm.
radiation. Anybody that comes to me, I'm not a physician, but I don't hesitate to tell them. I said, don't do it. Don't take chemo. Don't take radiation.
Is it are they are you s do they not destroy the cancer cells, or is it just that it
Oh,
leaves
sure,
some of them and then they
sure.
kind of have a field
Cancer
day
cells are
because
wimpy.
Yeah,
Every
yeah.
cancer cells. That's why it takes decades for solid tumors to appear. All right.
Mhm.
Almost most cancers always go away on their own. They're wimpy as they can be. Even lethal cancers are wimpy. That's why, when you get Coley's toxin or something like that, and you raise the temperature in your body, the cancer cells die out really, really fast. Your normal cells don't even die, but the cancer cells can't tolerate it. They're wimpy. Uh, and now, tell me your question again. I've already lost
Oh, just
track.
because why I was saying, you were saying chemotherapy should never, radiation we should never, and I
Oh,
was
yeah, yeah.
wondering, are you saying they're ineffective or just that they're
They're
just working on.
carcinogenic, and they kill cancer because the cancer cells are very, very wimpy.
Mhm,
It doesn't take much to kill cancer cells. It's very easy to do.
mhm,
Fever will do it, you know? And I got look at my hands. Up here is your normal health of your normal cells in your body. Down here is cancer cells. This is and up here is high, uh, good quality cells, bad quality cells.
mhm.
And when you take, a fever or chemotherapy or anything, these guys start going down real fast, you know? And the toxic stuff like chemo and everything, not only do the cancer cells go down, but your health goes down too.
Mm.
And then they have to take you off of chemo and radiation before they kill you. Can't kill all the cancer cells without killing the patient. Get rid of enough of them that you can't see, but still leaves millions of them around anyway. And now when you start getting better, remember you're not up here anymore. You had all the chemo, the radiation. It's depressing your health.
Mhm.
Now guess what? When you start coming back, here's the difference between your health and the cancer. And this cancer is resistant to all chemotherapy and radiation. You know why?
'Cause it lived
Because they
through it,
were the
yeah.
survivors.
Yeah,
All the
right.
easy ones were removed.
I see, so you kind of it's
You
the
know,
Darwinian, yeah, kinda like
yeah,
surviving, yeah,
they're there. And that's why cancer when cancer comes back really, really fast, and it's
yeah.
almost always lethal. And people have
Yeah,
been seeing this for decades. The oncologists see this. They know it. Most oncologists won't take chemo or radiation if they had cancer.
huh, it's interesting.
They see
Um,
it with their own eyes, you know?
are there any institutions sort of in the greater scientific or medical world that you do trust? Or are there do you see good guys in this?
Oh, they're always good individuals.
Mm,
I don't know of any good institution. That doesn't mean there aren't any. But the
right.
bigger the institution, the more corrupt it is. The more money that's going into it, anything that has to do with the government, I don't trust it. Anything that has to do with big money, I don't trust it. I
Yeah.
don't take drugs. I don't go to doctors. And
No.
and I haven't done for decades.
Mm. What's your regimen? How do you stay healthy?
I don't go to doctors and I don't take drugs. And we have a garden here. My
Yeah.
wife grows stuff. It's delicious. Only eat organic. Plus, my generation is to this day the healthiest generation of Americans because we didn't get all those vaccines. We didn't get all those poisons. Now we're killing the little kids before they're born or right after they're born. We're losing more and more of these little kids. And before COVID, before SARS-CoV-2 and all that, cancer was the third leading cause of death in the United States. Now it's number two.
Mm,
Yeah,
wow.
and with all these treatments, all these drugs, I mean, they're more toxic. They're destroying our own health. You see, remember that this thing they're reducing this distance between our health versus the cancer's health. You know,
Mhm. Yeah,
and you can't win that. Not by taking these things that keep this down, you know?
right. That makes sense. Well, I wanna recommend your book. Is it available where? How could people find it?
Well, I mean, maybe I should show them what it looks like
Oh,
as
yeah,
I
great
do it.
idea.
You do it such that it doesn't glare too much. The Outsider, it's all over. I mean, it's like Amazon.
All
It's
the
at
usual places. Okay, great. Yeah,
Trying Day, the publisher, they sell it too.
great.
The some bookstores do. I mean, it's a lot of places,
Yeah, and if people
and
have questions, can they find you online? Are you on social
yes,
media or
my
your website?
website.
Okay,
davidrasnick.com. I got over 40 years of information on that website. And my book's on the front of it on the homepage, a whole bunch of other stuff there. I got label for AIDS, for the history, all that, my history of the AIDS stuff, and it just goes back and back. And on cancer, the same thing. Tons of stuff on there, you
well,
know.
thank you so much for being here. The book is fascinating. It's as I mentioned early on. It's a balanced look at both your life as an outsider in the science and medical worlds, as well as your theories on cancer and aneuploidy and everything we've been talking about. So if people are interested in that at all, you should check it out. And you can find it on all the usual spots. Dr. Rasnick, I should say. Thank you so much for being. I always called you
It's
Dr. David, but Dr.
yeah.
Rasnick.
I prefer Dave,
Okay, Dave. Dave, thank you so much for being here. Really appreciate it, and thanks for your time.
It's a real pleasure. I enjoy it, and thank you for going to all the trouble to make this work.